All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show today. We are going to talk about the SARS-CoV-2 structure and the latest research that they have done. Many of the doctors and researchers have come together and done some research on its structure. It is fascinating and it is really interesting to look into it. It is my kind of science. So I thought that today we'll go over that. So let's start. I'm going to share my screen. So, of course, this is drbeen.com. This is the article based on which I have today's illustrations. And do tell me if you like the illustrations that I did today. So this is a beautiful article. With this article, there is this research paper as well by Dr. Lee, and that is about the SARS-CoV-2 spike protein and how it swivels or how it moves. And then there is another paper here, which is also, I believe, by Dr. Lee, and this is about the structure of the SARS-CoV-2 virus. And finally, there is a visual guide to the SARS-CoV-2 coronavirus, and it is the latest guide that we have. And it is, again, fascinating to look at various parts of the virus and how it works. So I would request you, encourage you to look at it. Can you imagine this one? This is the TMPRSS2 protein, which is going to cut this spike protein, and then this is the ACE2 enzyme. So beautiful structure here. So let's start. I'm going to be basing my flow or the structure on this article. So the very first thing in this article is about this structure of the SARS-CoV-2. So if you see this diagram here, let me just give the credit to the right people here. A cryo electron tomography image of SARS-CoV-2 viruses in gray with a computer reconstruction of one virus. Saili, Tsinghua University School of Life Sciences. I hope I'm pronouncing it correctly. Tsinghua or Tsinghua, I do not know what is the right pronunciation. But this is the diagram, and they have then computer generated this model here as well. So let us start. And of course, Luffy is today awake and is studying with us. So here what we did was, this is a cryo electron microscope. What it does is that it throws the electrons at a frozen sample of something, some tissue or viruses in this case, and then it creates layers. Imagine if we take a structure and then we shower that structure with cement. And so the end result will be that cemented structure will become sort of a statue, and then you take pictures of that, and that is a 3D model. So that is a similar thing that they did over here. So let's see what they did. So what they did was, Dr. Lee and his other research fellows, they first of all captured the virus, and then they added chemical substances that would make the virus inactive so that it's not harmful anymore. Then they freeze the virus. So they take this 1 quart of the fluids with the virus in them, and then they concentrate it down to 1 drop. And then what they do is they take that 1 drop and they freeze it. And they freeze it into and they have to rapidly freeze it. That freeze has to be occurring within part of a second. This is like flash freezing, which does on the food as well. It is a more sophisticated version of that. The reason to quickly freeze that drop of fluid is so that if it is frozen slowly, then the fluid would develop crystals, and those crystals can then pierce and rupture the coronavirus. So they do not want that. They want to see the virus intact, as you can see here in various. Do you see those little gray blobs here? These are actually coronaviruses. So they had to freeze them fast enough that the water does not develop crystals which would pierce and rupture the virus. And think about it, this is happening at the billionth of, I think, of an inch level. So once they have the virus frozen, then they bombard that virus with electrons. And the atoms in the virus would then cause the electrons to bounce off. And those bounced off electrons will be then picked up, and a computer would analyze those bounced electrons and construct a shape of the virus. So that is what they are doing. And look at this here. So what they do is, so Dr. Lee says that I thought I was the first guy in the world to see the virus in such good resolution. So what they did was, here is another image here. This is a virus that is, these are all cryo electron microscope images of in gray. And then what they have done is their computer generated a colored image. But this is an actual virus. It's not a simulated thing or illustrated thing. It is the virus which is then color coded by a computer. So fascinating stuff. And so if you see here, these are various parts, the procedures that they've done. And now if you continue here, the virus then looks like an egg's nest. So virus looks like a spherical structure, and inside the structure are the nuclear, the RNA, and RNA is covered by nucleocapsid proteins. You can say that these are covers, packaging on the RNA. So those packaged RNA pieces inside the virus, they look like eggs. So the structure looks like an egg's nest over here. So here, check this out. This is these yellow one are actually capsid proteins which are packaging around the RNA. And then this gray one is the virus membrane itself. And these red ones are the spike proteins on it. Now, for the spike protein, another researcher, Dr. Amaro, and his colleagues, they did a research on exactly what is the structure of the spike protein. And they came back with this message that it is really not a spike. A spike gives us an idea that it is something like, a pointed stick. So it is not really a spike. Instead, it is like a tulip. It is like a flower which has bloomed on the top, and it is cylindrical and short on the bottom. And the other thing that if you see here, look at this one. The spike protein swivels, moves around 3 hinges. So imagine our arm, we have a shoulder and the elbow and then the wrist. So these are the 3 hinges. And if shoulder joint, the elbow joint and the wrist all are making the stem of the spike protein, and the hand is the tulip part or the end part of the spike protein, that hand moves around on these 3 angles. And so this is the structure of the spike protein. And then these are the swivel angles around which the spike protein can move. So I want to show you that as well in a second. So give me 1 second. So if you see here, so the copyright for this one, I want to make sure that I'm not, this is only for educational purposes. I'm not taking their work. So this is again Sai Lee from Tsinghua University School of Life Sciences. Now continuing on, this is Dr. Amaro's work here. And if you see here, this is the spike protein. And look at how they are swiveling. So look at their attachment. Attachment is more like a stem. And then as the spike protein grows outward, it starts expanding. So it's really not a spike. It is a tulip-like structure. And that structure is kind of moving and swiveling. The benefit of moving is it is trying to find an ACE2 enzyme to bind. So it is kind of moving around and trying to find if it can capture an ACE2 enzyme and connect with it. And so if you see here, Soren von Bülow, Matheus Sikora, I hope I'm pronouncing them correctly, and Gerhard Hummer, Max Planck Institute Biophysics. So this is their simulation here. And kudos to the author of this article who has gone over all of these various researches, collected all of that data together that we are utilizing here. So this is by Carl Zimmer, and October 9 is when they have done is, this is New York Times. So back here, this is the spike protein. Then continuing on, then is a sugar shield. So then what happens is, if you look at this side, this diagram here, we have this bluish blob. And to explain that bluish blob, I'm going to actually bring the camera back to myself and use So imagine if I am the spike protein. And so what happens is that spike protein, imagine this is a candy, the cotton candy. So imagine this comforter here is a candy. So what the spike protein does is it kind of covers itself in this little candy here, or the sugar. It wraps itself in it and just pokes out one little piece, and that piece is sitting out there and trying to figure out where is the ACE enzyme and then bind with it. Now the question is, why is the virus trying to hide its spike protein in those sugar cotton candy wrappers? The reason for that is that the cotton candy, or these glycans, or these sugar molecules that are wrapped around the spike protein, they cause our immune system to be deceived. Our immune system is trying to, so somebody said Halloween scare. Yeah, so for the Halloween, this was a good show as well. So what happens is our immune system is kind of honing in onto the foreign proteins, and it is trying to figure out where are the foreign proteins that it can bind with. But the virus has done a clever thing, and that is that the protein itself is wrapped by glycans, which are sugars, and our immune system can actually be deceived by these sugars and not attack them. And if you see here, this little red thing is that little arm that the spike protein has, a little piece, a hook that spike protein has projecting outwards of those sugar wrappings, and through that it is trying to connect to an ACE2 enzyme. And then the whole protein is swiveling around and waving around to try to see where is an ACE2 enzyme that I can bind and connect with. So logic says, I got to show this. I just clicked on and saw you in a blanket, lol. Yes, definitely. So absolutely. So Caroline says glycated. Yes, so this spike protein, which is a protein, is wrapped in glycated. It is glycated, or it has glucose on it. So back here, and this is exactly correct. So M. Gregory says, smart suckers. Absolutely, these guys are smart. And Siddhartha says Halloween scare. Yes, Halloween scare. So back here. So what is happening now is that the spike protein is wrapped in the sugar, and those sugars are protecting it from our immune system, kind of deceiving our immune system to say, hey, there's nothing to see here. It's only candy. And then it has a tiny hook that it is trying to work with and connect to an ACE enzyme. Beautiful mechanism for the virus, not for us. Then another researcher, Joseph Puglisi, did one more part. So if you continue here, you would see the next work by Joseph. Do you see here? Look at this part. In here, you can see this little light blue thing is that hook on the spike protein that is bound to this ACE2 enzyme down here. So then I'm going to skip over the tangle loops and then go here to the jamming the machinery. This is another interesting thing. And the researcher here is Joseph Puglisi. And this is what I'm going to explain now. So what happens is that when the virus enters our cell and it is making it proteins, it needs to hijack our ribosomes. Our ribosomes are our machinery inside our cells that make our proteins. And what the virus does is, virus has a protein called NSP1. Virus makes the very first entry of the virus, and the very first protein generation from the virus produces a protein called NSP1. That NSP1 protein connects with our ribosome, which is our machinery to make proteins, and it has a secret location in the ribosome where it inserts the NSP1. And that converts our ribosome into a zombie ribosome. And our ribosome says, fine, I am at your service. I will now make more RNA proteins, virus proteins. So the virus itself hijacks our ribosome, or turns it into a zombie by a secret key it carries. And that key, when inserted into the ribosome, causes the ribosome to leave making our proteins and start making viral proteins. So that is weird. That how, and of course many viruses does that. There are so many viruses that arrive in our body, they live in there, they replicate in there, they hijack our systems. So it's nothing too unique here, but this virus has so many tricks with it that it eventually succeeds much more than the others. So this is the way the ribosomes work. So if you see here in this structure, if I can make it a little short. So if you see this pink area, that is our ribosome in which the virus has inserted its NSP1. Actually, this is called NSP. This is NSP1. NSP1 protein to hijack the ribosome. So if you see here, a ribosome with RNA in blue and with NSP1 in pink. So this is Christopher Lapointe, Stanford University School of Medicine, ribosome model by Angelita Simonetti et al., Cell Report, and Matthias Thomas et al., Science. So I want to make sure that the credit goes to them. Then I'm going to skip over this part as well, and this part too. And then I'm going to go to pores and tunnels. This, again, is a very interesting thing over here. So what's happening is, what you're seeing is, these are small spherical blobs, which they have taken, the scientists have taken the cross sections of. And remember, this is all actual real viruses pictures. These are not illustrations. These are just computer coded and colored, but this is electron microscope diagrams of the virus itself. So now what is happening is this. When the virus starts replicating in our body, so let's say this is the virus, this is our cell. The virus arrives in our cell, it starts getting into, the virus makes small, it asks our cell to provide it small vesicles or small pockets in which it can replicate. So here, this orangey thing is this a small vesicle in which the virus RNA is present, this green material. The question that the researchers were trying to answer was, they see the virus is being built, or the RNA are being built in this vesicle. The question was, how is this RNA coming out of this vesicle? And there were no pores, there were no outlets in the vesicle through which the RNA could come out. There was nothing. And so they were surprised, they were baffled that how come this virus comes out of this vesicle, the RNA. So they found out that the virus makes one more protein called NSP3 protein. That NSP3 protein is folded into the shape of a tube. So that is like if you take a paper and you fold that into a form of a tube. Then that tube is inserted into these vesicles here. And that creates a channel in the vesicle, that creates a tunnel in the vesicle, and through that tunnel, then the viral RNA comes out. What a wonderful thing that this virus is doing. So virus RNA is trapped inside the vesicles where it is being formed. Now that RNA needs to come out. To bring it out, it has a protein called NSP3, and that NSP3 protein folds into a tube. That tube is inserted in the vesicle. That creates a tunnel or a pore or a hole in the vesicle through which the RNA comes out. Beautiful thing, right? So then the last part here that I want to discuss, and just to explain this little diagram, let me stop it here. So let me explain what this diagram is doing. What they have seen here is that these are spherical blobs, and they have taken section of it. These green pieces are the viral RNA, and once that viral RNA is built, do you see these little red channels? These are NSP3 proteins. They get inserted into the blob, and through this the RNA comes out. So continuing on, then assembling the virus. So how does this virus get assembled? So again, this is electron microscope section. So imagine at the millionth of a piece of an inch, they are taking successive sections or slices of the virus, and then they are putting them together as a 3D structure. And so how does this work? So what happens is, and the researchers here are Dr. Gilad Felter et al. So imagine this little thing here, these little threads, they are viral RNAs. The problem that the virus needs to solve is that from one RNA, it needs to make one virus. It doesn't want many RNAs to be assembled in one virus. That will be useless. So what happens is it uses lava lamp physics. And you must have seen that in the lava lamps, the lava kind of rises up and then a blob pinches off, and that becomes one little bubble. Then some more lava rises up and another blob pinches off. That is exactly what happens. So imagine if there are threads of RNA. Around the threads and on the threads, there is another virus protein called nucleocapsid protein. That protein starts gluing itself onto the RNA. And when it glues on the RNA starts collapsing under it, and then finally it kind of pinches off like a bubble, and it becomes a one RNA with its nucleocapsid around it. Nucleocapsid is the packaging of the RNA. Then around that RNA is the membrane that is produced, and then that has the spike proteins and other things, and that is what makes one virus. So how clever of the virus that it actually evades wasting its RNA, and it knows, or it has processes to make one RNA into one virus instead of having multiple RNAs packaged in one virus. So if you see here, these are the threads, then they get collapsed with the nucleocapsid proteins into a small blob. That blob is then incorporated here, if you see, into this virus particle, around which is the membrane and the spike proteins and the N proteins and the other M proteins and so on, and that becomes a virus. So fascinating stuff. So if you see, that is that. And then if I continue on, this is an overall picture of the virus and how it works. And I would request you, for copyright reason, I don't want to go into detail there. And finally, the last point for today, this virus protein, the spike protein, do you see this kind of sapphire color blob and this orange color blob? These are the drugs and the antiviral things that can then get stuck onto the virus protein and make that thing unable to connect with the ACE2, and the virus cannot bind. So this is the approach for antivirals. So I thought that this was pretty interesting. One last piece here is the swiveling mechanism of the spike protein. So if you see here in this diagram, do you see this? Spike protein swivel mechanisms, and there are 3 hinges in it, and it kind of rotates around or swivels around those 3 hinges, and it is finding ACE2s. The same Dr. Lee, this is another link with his work and how the viral RNAs and nucleocapsids are present inside the virus and how the spike protein is present on top of it. So this is also a beautiful thing to look at. And finally, I loved this one as well. This is a visual guide to SARS-CoV-2, and here they have also discussed various pieces. So this is the discussion for today. Short and sweet, but I wanted us to kind of look deeper into the virus itself, look at the enemy, which is billions, probably tenths of millions of time of an inch, and how we have been able to now see the exact structure. And of course, we are going to defeat it soon as well. But this is what we have for today. So thank you very much for being here. Please make sure that you like, subscribe, and share. If you would like to support my work, there is a link to send donations in the description, and I would see you tomorrow.